All right, so welcome back. So this is part 6, and this is going to be our last part. And we're going to talk about some homeostatic set points. So this term, homeostasis, is an incredibly important term for us to understand in the context of anatomy and in the context of physiology. Our physiology largely is built around maintaining homeostasis. So what homeostasis means is essentially a state of steady conditions, okay, or relatively steady conditions that don't fluctuate too far in one direction or the other, right? So if we look here, homeostasis, we said the process of maintaining steady conditions within the human body. And so we know that in the body we have um, different ranges for different things. So whether it's glucose, whether it's sodium, whether it's blood pressure, whether it's heart rate, respiratory rate, whatever it may be, there's some type of um, set point and there's some type of range that exists that is considered normal, right? So for blood glucose, maybe we're anywhere from 70 or 75 up to 125, something like that, right? For sodium, maybe it's 135 to 145, right? If you're going for um, more education, you're going toward nursing or something like that, you're going to learn all these different um, set ranges for these different variables, right? So for glucose and sodium and potassium and calcium and all these different things, right? And so our goal is to keep those different things, glucose or sodium or whatever it may be, within a certain range, okay? And a homeostatic set point is just an established threshold that allows us to continue to um, functioning, physiologically functioning in a proper way. Okay, so our homeostasis is going to be regulated by what are called feedback loops. Okay, there are two different types of feedback loops. There are positive feedback loops and there are negative feedback loops. Okay, the majority of the ones we'll talk about are going to be what are called negative feedback loops. Okay, meaning you have a change in a um, some value of something, right? So say our blood glucose goes too high. Instead of being normally at 115, now we're at 180 or 250 or something, right? We're way too high. This is a stimulus, right? And the idea of a negative feedback loop is to bring that value back to normal. So whether we went too high and we need to bring it back down, or we went too low and we need to bring it back up, we're coming back to normal. Okay, we're trying to reestablish homeostasis. Okay, and there are different components of these homeostatic systems, whether they're negative feedback or whether they're positive feedback. Okay, so if you think of something like a thermostat in your house, right? You have you set it to a certain temperature, right? You set it to 70 degrees, you set it to 65 degrees, whatever it may be, and whenever the temperature, whenever it senses a change in temperature, and it goes too high, it'll bring it back down, right? So you have a sensor, you have to have a controller, you have to have some effector, right, that actually carries out the change, and then you can respond and you can bring your variable back to homeostasis. Okay, so we look at an example here of something like body temperature, right? The first thing we have in our this negative feedback, and again we're looking at negative feedback here, is you always have to have a stimulus. Okay, you have a homeostatic set range, right? So body temperature is maybe 98.6 plus or minus 1 or 2 degrees. Okay, you have to have some type of stimulus. You have to have something that alters your body temperature. Okay, so say we have something that causes our body temperature to increase. So instead of 98.6, now we're at 102.5 or something, right? We're running a fever. We have a high temperature. In order to detect that change in stimulus, you have to have a receptor, right? So that's one of our components. We have to have a receptor that detects that change in the variable, that detects that stimulus. Okay. We then have the receptor, which is part of our nervous system, right? So thermoreceptors in our skin send a signal, which in this case is a sensory signal, to our hypothalamus. Our hypothalamus is our internal thermostat, okay, that controls our internal body temperature, okay? That is the control center, all right? It then receives that signal and sends out its own efferent signal, okay, sends out a signal to some effector. Okay, the effector is what's actually going to bring about the change in the um, variable. So in this case, we have too high of a temperature. So the effector is going to do something. It's going to affect the change to bring that temperature back down. Okay, so in this case, effectors are going to be sweat glands that are going to increase production of sweat and blood vessels that can dilate in the skin to help dissipate a little bit more heat. Okay, so both of these have to do with thermoregulation. They have a cooling effect. We have increased heat loss, right? So evaporative cooling whenever the air brushes over our skin with the sweat and takes heat away with it. This is going to lower our body temperature back down. Body temperature returns to set point, so we have some type of response, and now we're back at normal body temperature. Okay, so we're back at homeostasis until this process happens again, and we can continue again, right? So if you look at the example of an actual thermostat, right? So we set it to 60 and it falls below 60. We have some type of receptor that detects it. It detects 60 degrees. It sends a signal. So just like we had a signal here, we have a signal going to our control center, which is the thermostat, which is set to 68. So this is now too low, falls to 60, we're set at 68. And this control center will then send out another signal. So in this case, to turn the heater on, right? Because we're trying to warm the room back up. We warm the room back up until we get to 68. Homeostasis is restored, and we shut off the heater, right? We shut off the heater until the room temperature falls below again, and we drop back down. Okay. So an important thing to understand for these negative feedback loops is we're always going to be fluctuating. We're going to be within homeostasis. We're going to be outside of it. We're going to come back within it. We're going to drop a little bit low. We're going to come back to homeostasis again. So we're constantly fluctuating in and out of homeostasis, but the majority of the time we're going to be within homeostasis. Okay, so we could have the same thing here for regulation of body temperature. You can also have the same thing for regulation of blood glucose or blood sugar levels, right? So we'll talk about this in A&P2, so in the next course in your sequence. But when blood glucose levels get too high, we're going to stimulate the beta cells in our pancreas to release something called insulin. Insulin is going to stimulate the uptake of glucose into the cells. This brings the blood glucose level back down until it's normal, and then maybe it drops too low, and then we have another hormone called glucagon that will then release more glucose to bring it back up, and then we go too high, and we have this constant cycle. Okay, so this negative feedback loop, we're returning to a homeostatic set point. Okay, we're getting back to homeostasis. Okay, so if we look here at an actual definition of negative feedback, we are fluctuating around a set point, and we're going in the opposite direction of the stimulus. So the stimulus causes our blood glucose to go up. We're going to try to bring it back down. Some stimulus causes our body temperature to drop. We're going to do something to bring it back up again. Okay, so we are fluctuating in this range. So this dotted line up here to this dotted line down here, this is all kind of a normal range. Okay, and so we're going to constantly be fluctuating above this normal point and below this normal point, and at times we may get outside of homeostasis. Okay, so it's a balancing act. All right. So this is a video. I'm not going to play the video right now. All right. So the opposite of this is going to be positive feedback. Okay. So if negative feedback is trying to bring us back to homeostasis, we're trying to oppose the change in the opposite direction. Positive feedback is going to be exactly the opposite. Okay. And this is not as common in the body, but this is a feedback mechanism in which we're going to move further away from homeostasis. Okay, and that kind of sounds counterintuitive, but it makes sense whenever you think of it in the context in which it happens. Okay, so in this case, some stimulus occurs, and our effect or our response is to exert some type of effect that causes it to continue in that same direction. So in other words, the change or the stimulus that brings about the change is amplified. Okay, the way that this stops, so in a negative feedback, the feedback loop kind of stops once we get back to homeostasis. Well, in this case, if we're continuing to move further and further away from homeostasis, you have to have some major event happen in order for this feedback loop to cease. So in order for this to stop. Okay, so you have to have some type of climactic event. Okay, so the kind of classic example in anatomy and biology is the release of oxytocin during uterine contractions. Okay, so what you have happen is during the birthing process, you have the fetus in the uterus, right? And during contractions, the uterine contractions, the baby's head is pushed against the cervix. Okay, pushing the baby's head against the cervix, which is the opening of the uterus, is going to stretch the the smooth muscle tissue in the cervix, and that's going to activate stretch receptors. So these are mechanoreceptors. The stretching of these mechanoreceptors stimulates a signal to be sent back to the brain, to the posterior pituitary gland, where we store and release oxytocin. Okay, oxytocin is a very powerful uterine a stimulator to stimulate uterine contraction, right? So, baby's head is on cervix, stretches it, stimulates the release of oxytocin. Oxytocin comes to the uterus and causes the uterus to contract. Well, what happens when the uterus contracts? It pushes the baby's head against the cervix. What does that do to the stretch receptors? It stretches them. We release oxytocin. More oxytocin causes uterine contraction, pushes the baby's head against the cervix, more stretching. And so you get this continuous loop until you have some climactic event occurs. Well, think to yourself, what is the climactic event in this instance? Well, the climactic event is the contractions get so strong that you actually deliver the baby, right? You get rid of the fetus, you deliver it. There is no more pushing of the baby's head against the cervix. There's no more stretching of those stretch receptors. There's not as much release of oxytocin. There can still be some contractions after birth, but it will not be to the degree that you have during this positive feedback loop. Okay. So read this question, pause the video, and answer it, and I'm going to go ahead and show you the answer. So which of the following is an example of a positive feedback loop? Well, in this case, it's going to be D. Right? So a baby suckling at nipples leads to more milk production. This is following a similar sequence of events as the uterine contraction example that we just gave. Okay, most of these others are going to be a negative feedback loop. Okay, we're opposing the change. We have an elevated blood pressure. We're going to try to reduce it. We have a lack of oxygen, so we're going to increase breathing to increase oxygen. We have an elevated body temperature, so we're going to produce sweat to cool and decrease the body temperature. Okay. All right. So these last 5 or 6 slides here are going to talk about. We're going to discuss some ideas of cells and some ideas of fluid movement in and out of cells that will be important in later lectures and important in AP2. Okay. So when we talk about Chapter 2 in our next lecture, we're going to talk about cells. And we have 2 compartments related to our cells. We have the cellular compartment inside the cell, and there's a compartment outside the cell. Okay. If the fluid is inside the cell, in that intracellular compartment, we call it intracellular fluid. If it is outside the cell, we call it extracellular fluid. Okay. If we have some type of disruption of this fluid balance, we can have some type of dehydration. We can have imbalance of ions, right? So you have to have a certain concentration of ions in the cells and outside of the cells in order for them to function properly. And we'll talk about this when we do our action potential lecture and when we do our skeletal muscle contraction. And if this is prolonged enough, you can actually lead to cell death or necrosis. Okay, so our fluid is going to move by something called osmosis. So we're going to talk about osmosis in Chapter 2. We're also going to talk about it in lab a little bit as well. Okay, and the movement of fluid during osmosis is determined by the concentrations of these ions in their relative compartments. So we'll see that fluid moves to where there is a higher concentration, okay, in an attempt to decrease that compartment's concentrations. Okay, so here we can see extracellular fluid outside the cell, so the plasma in the blood, and the interstitial fluid between adjacent cells, and then any fluid within the cell itself is termed intracellular fluid. All right, so we have different mechanisms that can cause fluid to move. Okay, we have one called hydrostatic pressure. We have one called osmotic pressure, and these will become important when we talk about filtration at the level of the capillary in AMP2, and when you talk about it at the level of the glomerulus in the kidney and the nephron, also in AMP2. Okay, so one pressure or one force is called hydrostatic pressure. Okay, well the term hydro is referring to pressure, and so I'm sorry, hydro is referring to water, excuse me, or fluid, and so this is the pressure exerted by the fluid due to the volume of that liquid on the wall of the vessel. Okay, so in your blood vessels, in your capillaries, you have blood, right? You have the fluid, and that fluid is exerting a pressure, okay, that is promoting filtration. Okay, so the important thing about this hydrostatic pressure in the context of capillary filtration, in the context of filtration at the glomerulus, is that hydrostatic pressure is promoting filtration. It's promoting fluid leaving the vessel. This osmotic pressure, this colloid osmotic pressure, is doing exactly the opposite. Okay, it is opposing filtration. It's promoting reabsorption, and it's trying to hold on to fluid, hold on to water, and it's due to the presence of large proteins like albumin that's made in the liver that's helping to hold on to the fluid, okay, and keep it within the vessel. So it's opposing filtration, right? So on the arterial end of a blood vessel, as we'll see in the next slide, there's a high hydrostatic pressure, low osmotic pressure, and this forces things out, or it promotes filtration, okay? On the venous end, or the kind of deoxygenated end of the capillary, you have a low hydrostatic pressure and a high osmotic pressure. In this case, this would keep fluid in or draw fluid back into the blood vessel or into the capillary. So if we look here, this is the arterial end, this is the venous end. Here we have a high hydrostatic pressure. We're promoting filtration. We're taking lots of fluid out here, out of the capillary. Here at the venous end, we have a high osmotic pressure, which is drawing fluid back in due to these proteins, like we just said, and this is promoting reabsorption. Okay. Now, if we have a lack of reabsorption, some of this fluid will go into what are called lymphatic vessels. And we talked about the lymphatic system in a previous part of this lecture. Okay, so fluid can go out, it can go back into the blood vessel, it can go into the lymphatic. If it doesn't go back into the blood vessel, back into the capillary, it doesn't go back into the lymphatic vessel, it builds up in this interstitial space, which interstitial just means in between the tissues or in between the cells, and it can actually build up, and this is what's referred to as edema, right? So you've likely heard of edema, maybe pitting edema when people have their legs maybe really swollen. This is due to an increase in the amount of fluid that they have that cannot be reabsorbed back into the blood, and it cannot be reabsorbed or absorbed into the lymphatic vessel. Okay. All right. So if you think of something like this, if someone becomes dehydrated, they can have problems thermoregulating, and so this is having to do with our homeostasis that we talked about earlier. And so their set point may be, or their specific body temperature may be off or out of their particular set point or set range, and so they would need to be able to control that. They need to be able to bring their body temperature back into homeostasis, right? However, if they're dehydrated due to the lack of fluids, like we just talked about in the previous couple of chapters, it can make it difficult to produce sweat, right? So sweat is dependent on fluid. Sweating is one of the mechanisms by which we can thermoregulate whenever we are overheating. Okay, so answer this last question. So most of our body's fluid is in which physiological compartment? So pause the video, think about it, and I'm going to go ahead and answer. So this is A. So this is intracellular. Okay. So if we look back here at this slide here, roughly 60% of our fluid is in the intracellular space, so in the form of cytosol, and about 40% is either interstitial fluid, plasma, or some other type of fluid, maybe like cerebrospinal fluid or something like that. Okay. So the majority of it is intracellular fluid. All right. So that ends this lecture. There are two kind of ending slides here. These are just additional resources that you may purchase if you want to help you in this course. So there are these visual analogy guides to human anatomy and physiology that have helpful ways to remember different anatomical structures and different physiological concepts. There's also a chemistry one if you need assistance with some chemistry concepts. And then there's a coloring guide, which can actually be very helpful when learning anatomy. There's I forget if it's a web page or a YouTube channel or whatnot, but it's called Draw It to Know It or Draw It to Learn It, and they really promote being hands-on with drawing and shading in an attempt to learn anatomical structures. So use these. There are other resources posted in the Blackboard course for you to look at, and you may use those if you think that they would help you. All right. So we are finished with chapter 1 with this lecture. If you have any questions at all, please email me. Please get in contact with me, and I will be happy to help. All right. I'll see you in the next lecture.